wonderful champions. Am I audible? Am I visible? Hello, hello, hello. So make sure you share it with all your friends who have given biology examination, because now we are going to do, what do you say, what do you call that? The paper discussion, exactly. And then only we will start, okay? Otherwise there will be children who will say that, ma'am, this question, that question. So it's better we start off together. Perfect, let's go. So, hello. I really hope all your examinations went on really well. I could get to see at least some of the children out there in today's paper. Start, please. Okay, perfect. In artificial hybridization approach, if the female parent produces unisexual flower, there is no need of, if the female plant is producing unisexual flowers, Baba, there is no need of what? Emasculation. Emasculation process is not needed. Right? Emasculation process is not going to be needed over here. Why? Emasculation means removal of the male parts. So we do not have to see that. Okay? Is it? The paper didn't go fine? What about others? What I believe, what I saw, what children told, it went really well. Yes, ma'am, 90 plus. Ma'am, full fire, ma'am. Exactly. So I think it was nice. Perfect. Thank you for keeping me motivated. I was in a doubt. Okay. 43rd question we will solve together, okay? Okay, fine. Emasculation is the first answer. The cells that secrete androgens are what? Sertoli cells, Leydig cells, germ cells, spermatogonia. What do you think is the answer for this particular question? This particular question. Cells that secrete androgens, Baba, these are all basic things that we discovered. It was known as Leydig cells. Leydig cells are the one that basically is going to produce the androgens out there. Now, sexually transmitted infection that can be completely curable is, so I told you everything that starts with H, for example, HIV, AIDS, okay, hepatitis B, genital herpes, all of them are not completely curable. You just have one thing, and that is gonorrhea, which is going to be completely curable. Okay, rest everything is not. So that is the question. Now identify the correct match. XO male grasshopper, ZW male bird, XX male Drosophila, ZZ female bird. My dear children, over here XO is the answer for this particular question, that is A. XO is the male grasshopper, ZW is basically the female bird, ZZ is the male bird, XX should be female Drosophila, not the male Drosophila, which is why your answer for this particular question is going to be A. Fourth answer is A. Perfect. So I'm seeing answers flowing up there, and that to correct. Perfect. Next question. A nitrogenous base is linked to the OH of the first carbon pentose sugar through N-glycosidic linkage to form a nucleoside. This is the thing. So over here, when you talk about the structure of the ribose sugar, the pentose sugar, on the first carbon, Baba, this is the first carbon. What do you have? You had a nitrogenous base, and what do you call this bond as? You call this bond as N-glycosidic bond. That is being asked, and the whole structure that is being formed over here, we will call this as a nucleoside, right? Will you call this as a nucleoside? Exactly. If phosphate is not there, I'll call this as a nucleoside. Now, two nucleosides are linked through 3,5-phosphodiester linkage to form a dinucleoside. Two of them are linked together to form basically, Baba, to form a dinucleoside. The answer is wrong, because over here, if at all you have some other ribose sugar and they are linked through 3,5-phosphate bond, basically it means that they are dinucleotides, not sites, okay? So that is one very important thing. Sarita, ma'am, because of you, I'll get above 60. Thank you so much. Thank you so much, love, for coming and commenting that as well. It means a lot. Okay, so now for your answer, this particular statement, I say statement 1 is correct and statement 2 is incorrect. Statement 1 is correct, but statement 2 is what? Incorrect. Your answer is going to be C for this particular question. I hope this is clear. Yes? Yes. Perfect. Next, correct sequence of evolution of animal groups according to geological time scale. I told you, I gave you the formula to remember. Farm. Farm is what? Fishes, amphibians, reptiles, mammals. Fishes, amphibians, reptile, mammals. Sufia, because of you, I'll get 50 plus. Oh my God. Thank you, dear. Perfect. So your answer for this question is going to be D, fishes, amphibians, reptiles, mammals. Exactly, Anjana. Farm. Perfect. Going ahead, the molecules trapped in the lymph nodes. Morning, we discussed in the model question paper live also. The molecules trapped in the lymph nodes responsible for the activation of lymphocytes present there and causes the immune response are the one that is going to be, increasing the immune response out there. What is that? It is going to be, Baba, none other than the antigen. Exactly. So your answer for this question is going to be C. Okay, it is going to be C. Abhishek says, ma'am, I'll get 70 on 70 pakka. Perfect. Awesome, awesome. Alaina, yes, dear, tell me. I hope I really hope all your examinations went well. This question we discussed it yesterday. The plants with hallucinogenic properties are atropa and cannabis, datura and atropa, datura and cannabis, atropa and papaver. The answer for this is what? Datura and atropa, B option, because datura, atropa belladonna, and only one more thing, that is erythroxylum coca. All these things are basically this thing, okay, the one that gives you hallucinogenic properties. Prajwal, Miss Yela Cupiniv, expect Madhiri Hagu, thank you very much. Thank you so much, dear. Sarita, because of you, I'll get, okay, thank you so much, guys. It means a lot. Okay, just messed in 4 MCQs, still certify. Yes. Perfect. Now going ahead, ma'am, I took additional also. Perfect, perfect, awesome. Next thing, match the list 1 with list 2. List 1 is here, list 2 is over here. You have to match each of them. So rhizobium is there, clostridium is there, aspergillus is there, methanogens is there. I told you rhizobium which is there, it is what? It is nitrogen fixing bacteria. Methanogens which is there, Baba, last one, it is found in gobar gas, or basically it is producing gobar gas, that is your methane and everything else, but biogas. Clostridium which is there, it forms butyric acid. Aspergillus which is there, it forms the citric acid. So your answer for this question is going to be A goes with 3, and here is the answer. Your answer is going to be C option. Perfect. Perfect. Awesome. Yes, and 90 people are watching this video. I hope, my dear children, your paper really went well. And do not forget to hit the like button and share with your friends also, okay? Now, which of the following is the correct representation of primer annealing step in PCR? We did it yesterday. What is the correct step of primer annealing step? I already told you, only if the primer is attaching at the 3' end, it will be correct, right? It is going to be correct. So, over here, my dear children, the first option, one side 3' end, but 1' 5' end, wrong. Here, at the 3' end, okay, and here at the 5' end, wrong. Next one also you have at 5' end, wrong. The last one is the correct answer. D is your correct answer. Here also the 3' end, here also the 3' end. What do you have? You have the primer being attached. Ma'am, thank you so much. 60 on 60. Sorry, 80 on 80. Sayada, perfect. So happy. So happy for you guys. Okay, next thing. Identify the incorrect statement. Now over here, we learned it yesterday, but still I am telling you, listen to me carefully. Where children might have missed is here. First letter of the restriction enzyme is derived from the genus of the bacterium. Correct. For example, E. co R1. First is from the genus. Correct. Escherichia. Second and the third letter of the restriction enzyme are derived from the species. Coli, CO, right? Fourth letter of the restriction enzyme is derived from the strain of the bacteria. I told you our strain is there. So basically, that is also correct. The Roman letter after the strain indicates the Roman origin of bacterium. No, it just indicates the origin of discovery. There is no Roman origin of bacterium, right? So Baba, over here, the answer for this particular question would be D. This is the incorrect statement. You have been asked to find the incorrect statement. This one is the incorrect statement. Rupa Shri, ma'am, me also 70 on 70. Perfect. Lakshmi says 90 on 20 in the MCQs. Is it? Which one went wrong? But anyways, perfect. Super happy for you. Perfect. Haji Ali, ma'am, 20 on 20 in part A. Perfect, guys. Okay. 10th is C, not D. No. 10th answer, it is going to be D only. There is no doubt in that. 10th is going to be D. Until now, all the answers which are given are correct. Now, the corn borers are controlled by protein encoded by the gene. The corn borers are controlled by Cry1AB. The Cry1AB, the answer for this question is going to be 12th. It is going to be 12th. It is going to be 12th. Okay? Ma'am, shall I say something? I have attempted all 2-3 marks, including options, but MCQs maybe. Okay? MCQs maybe you're losing 1 or 2 marks. It's okay. It happens, guys, it happens, but at least you gave your best, right? At least you gave your best, and that is something that is super, super important. Yeah? Yes. Now, herbivores in a broad ecological context not vary from. Herbivores, my dear children, in the broad ecological context would be what? Commensals, predators, parasites or producers. Basically, they are acting like a predator. Why? Because they are going to eat the prey, which is basically the plants out there. Ma'am, I got Haji, 20 on 20 in part, but I have doubt on last main question. It was not easy. Maybe I lose marks in the last main. Okay, we'll see. We'll see. Anyway, you have options to write. I hope you have written the answers which were given in the options. Yes? Let's go ahead. Third letter is strained. No, Baba. Third letter is this letter. No. How can it be strained? The fourth letter is strained. No, Baba. See. Guys. This is important. This is important. All correct, ma'am. Ram. Perfect, dear. Now, the next one that we have is, okay, we have to move ahead. Dead plants remain such as leaves, bark, flowers constitute what? Whenever something dead and decaying is there, basically we'll call them as detritus. What do you call this as? Detritus. Ma'am, all correct. Perfect. Yes. Okay. I hope that works. Now, next thing, an estimate about 7 million global species diversity was made by, again, in the live we discussed all these things. It was, it was made by, estimate what made by Robert May. Robert May was the person who said that it is going to be 7 million. Agreed? Done? Robert May, exactly, Trisha. 4 marks gone, is it? Perfect. Let's go ahead with the fill in the blanks then. Is it fine? 68 plus out of 70. Perfect. Awesome. Seeing that makes me really happy. You know what? I was literally happy seeing the paper. 20 and 20. Mikey says 20 and 20. Okay, perfect. Didi Konde, oh my God, everyone. And Shri, it happens. Okay. Let's go ahead then, guys. So the initial few days of milk produced after parturition is known as what? Colostrum. 16th answer would be colostrum. The Homo erectus had a brain size of, Homo erectus, Baba, it had a brain size of 900 cc. It was more than Homo habilis. Right? It was more than Homo habilis. So over here, your answer for this question is going to be 900 cc, 17th question. Now, human immunodeficiency virus replicates in, Baba, macrophages. So your answer is going to be macrophages. Now, next one, a small amount of curd added to fresh milk. If at all we are adding small amount of curd to the fresh milk, what will be your answer? Okay, what will be your answer over here? It is going to be where? Inoculum. It is known as inoculum. 19th answer is inoculum. Done. Now, next thing that we have, based on currently available species inventories, 70% of species recorded are, 70% of species which are recorded, they are going to be animals. This is also something that we discussed. I think most of the things here and there we have discussed, and I am really saying it is, Ishant, ma'am, I am a NEET aspirant, writing all the things, losing marks in MCQ is very painful. I lose 3 marks. You know what, it happens, and now it's done, right? But you have still NEET examination which is going to come. Make sure you give the best. Okay, Trisha, ma'am, your hard work for us made us today's paper easy for us. Thank you so much. Thank you, sir. All the questions in the last main were difficult. Is that so? I don't think so, but yes. Haji Ali, ma'am, it was possible because of your classes. Thank you so much, JJ ma'am, and lots of love. Thank you so much, guys. Lots of love to you too. And Banvika says that 70 and 70 all because of you, man. Thank you so much, guys. Thank you so much, Trisha, your hard work. Thank you. Sarita, I remember you and your lecture in the examination. It was really helpful, ma'am. Thank you so much, Prachi also. Everyone. I hope that I helped you in some ways possible. Some people are saying that the paper was okay, and I feel little in dilemma. The paper was not actually difficult. It was actually easy. So, yes. Yeah? Yes. Ma'am, 13th should be D. No, 13th is what, Baba? Let me see. 13th, which will be there. No. They herbivores. You're talking about herbivores, the ones that eat the plants. How can they be producers? They are eating something. They're heterotrophs, right? They are going to be heterotrophs. Okay? So, Sophie, it was really helpful. Thank you so much. Okay, perfect. So, let's go ahead. All the MCQs and the FIBs, that is fill in the blanks, are clear. Can I go ahead with the next set of questions out there? Will it be okay? Okay, fine. Next thing, draw a label diagram of matured two-celled pollen grain. We know that we drew it today morning also, okay, and we have drawn it earlier also. That is basically you're talking about the male gametophyte. Make it properly. Make sure the label was also labeled. It was labeled as well. Sanjana, I met Sanjana today. Hi ma'am, paper was easy. I hope you're the same Sanjana. Paper was easy because you teached in an easy way. Thank you so much, dear. It means a lot. Okay, perfect. Going ahead, how is ZIFT different from IUI? ZIFT is basically what? Zygote intra-fallopian transfer. Azad, just a second. Please recheck. Azad is saying, please recheck. Okay, so I'll recheck for you once more. Listen to me very carefully. Eco Arvind, this is the first letter, second letter, third letter, fourth letter, right? Eco is there. First letter is for the genus name. Second and third letter is for the species name. And the fourth letter, Baba, this is the fourth one that is derived from the strain. That is the strain only. So where, it's not wrong, dear. Ma'am, 69, but I didn't study the MQP, is it? Okay, perfect. Yeah. So ZIFT is basically zygote intra-fallopian transfer, where we are transferring the zygote directly inside the fallopian tube, or probably it can be an embryo up to 8 blastomeres, which is put in the fallopian tube. That is ZIFT. How it is different from IUI? IUI is what? Intrauterine insemination, where inside the uterus you are basically passing on the sperms out there. Yes. So that is the difference that we have. Ma'am, what KCET and NEET classes your channel having online classes? Ma'am, your channel will be having definitely online classes out there. And also we have launched so many courses. Make sure you go through it. It is definitely going to help you. Muhammad says, ma'am, definitely more than 65 just because of your health. Thank you so much. Thank you so much, dear. Trisha, ma'am, I literally remember you teaching the concepts while writing the answer. Oh, that's so sweet of you. Perfect. Okay, sketch the pedigree symbols, that is sex unspecified. When sex is not specified, you make it like this. When you want to show mating, you make it like this. Plus, over here you have a square, circle, and then one line in between. Okay, ma'am, I lost in 15th question. Okay, that is the one question that you missed, right? It happens. Okay. So for KCET, dear, we have launched Abhyas KCET test series, that is there. Apart from that, there is KC30, which is there, a crash course. If at all you're interested, make sure you take it up. It will be really, really good for your preparation. And previous year, it worked really well. So yes, Vinita, ma'am, 67 plus, awesome. Now, mention 4 properties of genetic material. What are the 4 properties? First, it should be able to replicate. Second, it should be able to mutate at a specific rate. And yes, it should be stable. That is one feature. And it should be able to express the genes in the form of Mendelian inheritance. So these are the few things that is there. Okay, for MCQ questions, you're not going to get half marks. In the case of this, yes, you might probably. Okay, now what are homologous organs? The organs basically which have underwent divergent evolution. They are structurally similar, but functionally they are not similar. Okay, so functionally they are not going to be similar. So 25th answer is, and you have to give an example. You can give the example as you give the example as anything. For example, you have 4 limbs of cheetah, 4 limbs of birds, and everything out there. So ma'am, full auto out in MCQs, perfect. Ma'am, 23, first I made circle and then square. You usually make square and then circle. That is how it goes. Okay, that is how it usually goes, dear children. And by the way, I hope you guys are, are clicking on the like button as well of this video, if at all you think that it is helpful for you. Now, antibiotics acts as against life as well as pro-life to the organism. Against life, basically, you're talking about the bacteria and all which it is being killed. That is against life for that reason. And pro-life means it is going to help us. In our case, in the case of human beings, it is beneficial for us. That is why it is known as pro-life. Now, if you think that basically this particular question was tricky for you, obviously you have an option to choose and go with the next one. List the evil quartet. We discussed that. Habitat loss, overexploitation, and invasive species, and then finally we have co-evolution. These are the 4 points that is out there for habitat loss, that is evil quartet. Now we go ahead. So basically with what we have learned, I am literally assuming that every one of you, so 7 questions are there, and out of that 1, 2, 3, 4. and 5. Exactly 5 questions was something that we probably might be knowing very easily. They are all easy questions, right? So yes, now the next thing that we have is part C. Draw a label diagram of a monocot embryo and label the three parts. I made you draw that, okay, several times. I have given you in that also, so make sure. I hope you had watched it. Yes, so 3 points in 25th. Okay, you have wrote only 3 points in the 25th one. Okay, it works in the 25th one. Okay, now let's go ahead with the next thing that is there. And yeah, so diagram 1 is clear. Now what is placenta? Mention 2 function of placenta. Placenta is what? Basically a temporary link that is there between the fetus as well as the mother. And over here you have the role of chorionic villi and everything. Chorionic villi along with the uterine tissue makes up the placenta. And then you go ahead explaining the function of the placenta. It is a temporary endocrine gland. It helps in the excretion, basically removal of waste materials. It helps in what? Absorption of, it helps in exchange of gases and everything out there. There are a few points which are there. You just have to go ahead with it. Prabhavati says, ma'am, 65 plus, thank you. Okay, thank you so much for coming and commenting about that. So happy. Perfect. Okay, yes. Now explain the modes of action of intrauterine device. What is the modes of actions of intrauterine device? Intrauterine device, you have to tell that basically there are 3 different types out there. One does phagocytosis, that is basically the first one non-medicated IUD. Second one basically messes with the motility of the sperm and the fertility of the sperm. And the third one basically it does not allow ovulation and implantation to happen, all these things. Okay, so this is very, very important. Ma'am, please teach biology for KCET and NEET on YouTube. Please, ma'am, we love you and your teaching a lot. Definitely would be doing that. Ma'am, I wrote CUT is which prevents pregnancy by releasing copper. Is this correct, ma'am? Yes, but you have to go ahead and explain more about all the, you have to tell the modes, no? So different modes which are there, you have to explain about that. So as I told you, different IUDs have different modes of action. Go ahead and explain about that, then only you will be getting the marks, and or else depends upon the teacher who probably is going to check. Okay, I wrote 27th in my own words. 27th in your own words. Okay, it works, but I hope you have mentioned this term somewhere. That will be really good. Lakshmi says 96 out of 100. Perfect, dear. Okay, state Hardy-Weinberg principle. Mention the 4 factors that affect the Hardy-Weinberg equilibrium. We studied it together. Okay, Hardy-Weinberg principle says that frequency of alleles are going to be the same in every single generation. Sorry, and basically the factors that are affecting is anything. It can be what do you say? It can be gene flow, it can be gene migration, it can be genetic recombination, it can be natural selection, genetic drift. All these reasons are there. You just have to go ahead with that. Yeah? Yes? Perfect. So there's a few things that is out there. Apart from that, what do you have? Can you clarify? 13th, ma'am. Again, 13th question. Why does everyone have a doubt with 13th question? Yes, I can. 13th. Where is it? 13th. Herbivores. In a broad ecological context, not very much different from herbivores are the ones that eat the plants. So aren't they acting like predators? They are the predator and prey is what the plants. Okay, so your answer for that question is going to be B. Perfect. So simple beauty, I hope that is sorted. And now going ahead with the next thing that is there. So Hardy-Weinberg is sorted, we discussed about it. And the next thing that we have over here, mention the disease caused by following pathogens. I told you that these organisms might be there. So yes, ma'am, do people correct strictly. We're not sure about that. Hopefully they will be doing good to you. Okay, do not be worried. Now, fine. 43rd, yes. Ascaris basically is caused by ascariasis and mention that is, Ascaris causes ascariasis, Haemophilus influenzae causes, pneumonia, trichophyton causes, that particular disease which you call as ringworm disease. So these are things are there, right? So these are a few set of questions which are there. Next thing, Streptococcus, Trichoderma polysporum, Monascus purpureus. Name the bioactive molecules that are produced by, so they act in an opposite way, you have to tell. So Streptococcus produces streptokinase, Trichoderma polysporum produces what, Baba? Cyclosporin A, and Monascus purpureus produces what? Statin. So I told you this easy peasy question, ma'am. Exactly, easy peasy question, right? So very scoring thing that is there. Sketch and label the diagram of pyramid of biomass in a terrestrial ecosystem. We studied this also. So you have to make the pyramid like that in a terrestrial ecosystem. It looks like this. Mention primary producer, primary consumer, secondary consumer, tertiary consumer. Put the numbers, highest number over here, then low, then low, then low. Put the unit, dry weight. Okay, dry weight, kilogram. And I told you to write like this. I hope you have written like that. Yeah, these are the few things that is there. For Trichoderma, I wrote Cyclosporum, will I get full marks? For trichoderma, I wrote cyclosporin. That is cyclosporin, right, dear? So I'm not sure whether you're going to get full marks for that or not. Hard worker, ma'am. Thank you so much. Just from your classes, I scored A+. Thank you so much. Is that my new name? Hard worker, ma'am. That's so sweet. Okay, mention for any 4 of the application. This is 5-marker question. Explain the formation of mature embryo sac from a functional megaspore. So you have to talk that one functional megaspore which is there. It is going to undergo what? It is undergoing mitotic division. How many times? Thrice it undergoes mitotic division. You get 8 nucleus at the end. You get 8 nucleus at the end. And then basically you have to take the help of a diagram. Okay, make the diagram of the embryo sac and take the help of that to basically give ahead the answers for it. Okay, so you then say that these nucleus which is there, they will be having cell wall around it and they will be arranged 3 at the top, you call them antipodal cells, 3 at the bottom, 2 of them are known as synergids, 1 is known as what? Egg cell. So all these basic things you have to tell. That's all. Okay. Pallavi, ma'am, I ask you something. That 10th question is that C option, because in model question paper, celebrity, it's okay. One question is wrong, right? That's okay. That's fine. 10th question, guys. 10th question. The answer is D for that. There is no doubt regarding it. I'm telling you again. And if you wait for some time, I can explain you once more as well. Yes, and then, okay. So that has been done. So you have to explain all of that, my dear children, and it's sorted. You have to make the embryo sac diagram as well. I hope you had done that. And draw a diagrammatic sectional view of human female reproductive system and label the 5 parts. I told you one question is going to come from this particular chapter. You are going to get your 10 marks from here. Your 5 marks from here. And obviously in the previous also you have got marks. So over here, your answer for this particular question is going to be what? Make the diagram of the female reproductive system properly. You'll get all the marks. Okay. Now, schematically represent the results of inheritance of incomplete dominance in Snapdragon. We discussed about it. Okay, what do you have? Okay, what do you have? Ma'am, 20 on 20. Love you, ma'am. Thank you. Love you too, dear. Okay, so over here, incomplete dominance, you have to explain with the help of capital R, small r, and when you cross them, you get capital R, small r. This is not completely dominant. This is going to be what? Incompletely dominant out there, which is why you get pink color. And then you go ahead and talk about everything else out there. Okay? So, yes. Okay, next thing, describe the regulation of lactose operon in the bacteria. Again, something that we discussed. Guys, you tell me, if at all you are someone who have attended my class, how can you feel that the paper is difficult? Because I'm literally saying, if at all you go back and look at it, okay, you look at the live, you are literally seeing that we have done these questions. Okay, we have done these questions and it's like really, really great that yes, exactly, Tara, ma'am. Everything came from your marathon. Love you, ma'am. Thank you so much for acknowledging that. So initially, two people came and said something. I was really sad. They said that it is not exactly what is there. So I was like sad, and now I feel really happy seeing all of you guys coming up and telling the answer. Right? 10th option is C. No, dear celebrity, go check your NCERT once more. The primer is attached at which side? 3 dash, okay, 3 dash side. It is in the picture, it is inverted and given to you. That does not mean that it will attach on that particular side only. Look at the 3 dash end. It is attached at 3 dash end. Now in the question paper, is it a 3 dash end or not? Look at that. I told you these type of questions could be coming. We solved it also. Yeah? Yes. Yes. Okay, next thing. Define contact inhibition and metastasis. Contact inhibition means cells when they come in contact, what is going to happen? Hello? No. My voice is not muted, Tara. Yes. I think increase the volume. Okay. Cells when they come in contact, they are not going to go ahead and divide. Okay. That is usual thing that is found. In the case of cancer cell, they lose this property, which is why the cells are going to keep on dividing, even if when they come in contact. Okay, and next thing that is there, metastasis. Metastasis is the most bad property, malignant property of cancerous cells, where basically these cells move from tissue and go out throughout the different parts of the body. They spread throughout the body. So this is very important. Okay, scientifically, ma'am, I have written sporopollen A instead of cyclosporin A. What shall I say? Okay, ma'am, 60 plus Ridha Khan and dynamic 55 plus. Perfect, guys. Good. Okay. Going ahead, okay, explain the treatment of cancer. So you have to go ahead with different, what do you call, you have chemotherapy, you have radiotherapy being done, and then you have, what do you call, this thing, immunotherapy, where alpha interferons, okay, are being given. All this thing you have to explain. So there is surgical method, there is radiotherapy where radiations are being given, there is chemotherapy where drugs are being given. Apart from that, you can also talk about what? The alpha interferons which are given as basically this thing they said. Hita, it is not according to textbook. Guys, you are looking in the wrong way. Please make sure you check your NCERT properly. Where is the primer attached? At the 3- end. The picture is given ulta over here in this particular question. And now you're believing, you have to check guys, do not do blunder. Okay, now write a note on the benefits of transgenic animals. We literally discussed about it as well. Transgenic animals, they are there for natural understanding of normal physiology and development. They are helping in understanding the different type of diseases which are there. They are basically helpful in understanding the vaccine safety testing, the chemical toxicity testing. Also, they help in biological products, for example, alpha-1 antitrypsin and also alpha-lactalbumin. Okay. Yes, I checked the textbook. 10 is D, 100%. Exactly, 10 is D. Okay. Why did they give ulta picture? That though I cannot say, but I told about the ulta picture also in one of my lives. I told that this is how it comes, so you should be very, very careful. So yes. Rosy cow, exactly, Stoner. So now, mention an equation to calculate the population density at time t+1. This is from your MQP directly. So you just have to say nt+1 is equals to nt, okay, plus what? You have your equation, that is birth plus immigration minus death plus emigration. This is the formula that you have to write. If at all you have written this, you might have got your marks out there. Okay, so I hope this is clear. 41st question. In equation, I wrote 2 times in equation because I had confusion. So will my correct answer be considered correctly? Dear, so usually if at all you have made a cut out there, depends. So children who get less marks, in that particular case, sometimes they think about it. They think that they want to give more marks and they do the easy checking away and they give that. But yes, I'm not sure about it, dear. Okay. Next thing, 42nd question. Now we are entering the different zone out there. I hope every one of you might be able to. So you had to write basically how many questions from this particular section. You have to write 4 of them. I hope you are able to make the embryo sac thing. You were able to do the female reproductive system thing. The incomplete dominance thing is there. Lack operon is there. Transgenic animals are there. All these things are easy things. It's more than 4 questions that you can do over here. So this is very, very, very important, right? So yeah. So, and I hope you did that. So your 4 questions are already been done. And now basically we go ahead with one question that we have to do from here. Any one question. Describe blood associated gene disorders linked to sex chromosome and chromosome number 16. Any idea? Blood associated gene disorders linked to sex chromosome. When you're talking about sex chromosome, basically you're talking about what? Hemophilia. Okay, blood disorder which is linked to sex chromosome, it is going to be hemophilia. So you have to talk about hemophilia, right? Kirti ma'am, promise ma'am, I started studying biology after chemistry exam, that is 4 days holiday. Given for math, I'll get 60 plus 70. I followed your all life. Thank you so much. You're most welcome, Kirti. That means a lot. Okay, so over here, your answer for this question is going to be hemophilia for sex chromosomes. And over here, your answer for this particular question is going to be thalassemia. It is going to be thalassemia. Fine. This is very important. Yes. And yeah, exactly. Alpha thalassemia. Alpha thalassemia, you have to mention that because there is beta thalassemia as well. So alpha thalassemia is the answer for that. Ma'am, I wrote only thalassemia. What happens, ma'am? You might have explained a bit about it, right? You might have explained about what type of thalassemias are there and things like that, right? If at all you have written it somewhere, at least they will be giving you some marks. Okay? Yes. I wrote 2 equations for, okay. If both are correct, why are you asking? Obviously, you'll get marks, no? Yes. Okay, next one that we have is refer to the schematic representation of DNA fingerprinting thoroughly and answer the given question out there. Okay, fine. So, how many short tandem repeats are shown in the 7th maternal and paternal chromosome of individuals A and B? Let's go ahead. So, here they have clearly mentioned how do you need to count the number of short tandem repeats. Here they are saying there are 11 of them. Now, come to this particular picture because that is our important thing, right? So, we have to calculate, this is the paternal chromosome, these two. Okay, this is of individual 1, this is of individual 2. Okay, individual B. This is the maternal chromosome, these two. Now, you have to count the tandem repeats which is out there. In individual 1, in the paternal chromosome, you have 1, 2, 3. Okay. In individual A, in the maternal chromosome, you have 1, 2, 3, 4. Done. Okay. Now come over here. What do you have? In individual number B, the short number of tandem repeats which is there in paternal chromosome is 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. 11 of them are there. Over here in this particular individual, in the maternal chromosome, 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 of them would be there. So this is how you need to write for the first answer. Now the next thing that is there, how many short tandem repeats are separated on the gel from individual A and B? So if at all you see over here, this is A, this is B, you have to just calculate the ones which are being spread out. So for individual A, 1, 2, 3, 4, 5, 6 of them are being spread out. Whereas for individual B, 1, 2, 3, 4, 5, 6. Again, this is also 6 of them which are being spread out. Okay, done? Yes. Okay, now going ahead, what do we have? Identify and mention the number of short tandem repeats that are common to both individuals A and B. Mention the numbers. So over here they have given you the numbers, right? Over here you have been given the numbers, so you have to identify according to that. So basically we can say the 5th one. Okay, the 5th one, the 7th one and the 8th one would be the one which is common. So here if at all you look my dear children, this thing is common, this thing is common and this thing is common. Now I know it is like a bit ulta seedha that is being put over here. They should have put it in a proper way, alignment, but they have not. And this is exactly taken from your NCERT. In your NCERT also it is like this only. Okay. Ma'am, I learned Monaco diagram in today's morning live. Thank you for the explanation, was awesome. Thank you, Pranava. Okay. I wrote only thalassemia 1 page. Khanu, I do not know how many marks at all, like segregation for that, probably 2.5, 2.5. Yeah. Yes. Okay. So that is the thing that is there. And yes. So these are the things, and now you have DNA from the crime scene matches with which of the individual. So if at all you see this particular DNA, this DNA matches with which of the individual, you can clearly see this and this matches. So we will say C and B are going to be the one which are matching out there. C and B are the ones which are matching out there. Okay. Ma'am, for B options I have written individual A6. For B option A6, okay, and A6 and B6. Yeah, correct. Yes, correct. Are you being asked about the total number? No, total is not being asked, so you do not have to write total number thing. Jetaram, thank you so much, ma'am. You're most welcome, dear. Okay. Perfect. Okay. Guys, going ahead with the next theme that is there. Is this done? No. You have one question. Yes. You have one question, and that is this one. Refer to the given diagram and answer the following question. Now, I hope that if at all you were not able to write the first question or the second question, you should be at least able to write this particular question. This stuff we have discussed so many times, Baba. This stuff we have discussed so many times, right? So, ma'am, getting 80 plus in this paper, it got bad. Good, definitely good. I don't like this question, ma'am. Yes, you don't. So, which enzymes can be used for insertional inactivation of selectable marker genes? So, if at all you want to insertionally inactivate the selectable marker genes as an ampicillin as well as tetracycline, I told you, you have PV1, PST1 or VAMH1 and SAL1. These are the things that you're going to use. Now, vectors should not have more than one cloning site for the same restriction enzyme. Why? I told you several times that if at all you are talking about a vector and you are saying that they have this particular enzyme has multiple cloning site, what will it lead to? It will lead to fragmentation, because this guy will make a cut over here, this guy will also make a cut over here, leading to what? Fragmentation of the DNA out there. That is why you will not use it, right? Yes. Now, what is the significance of ROP? ROP site, I told you, it forms the protein that is super important for the process of replication. Right? Yes. Now, which region of the plasmid controls the copy number? This one, ORI site, is the one that is going to control the copy number. Okay, I hope that is clear. And over here you have for the visually challenged children, you have different set of questions out there. So, yes. Sickle cell anemia, chromosome number 16, no. Sickle cell anemia is not affecting chromosome number 16, no, Baba. I just replayed your video in my live, in my mind, ma'am. Thank you so much. Ma'am, 44th question saved me. Biotech always saved me. Yes, that's why I always say that always have a backup. I always say that. Always have a backup. Do not look for the easier way out there. Right? Okay. So, one more very important thing before going ahead, and that is, ma'am, I'm getting 96. Perfect, dear Tanu. And yeah. So, once the live ends, please let me know in the comment section, were the videos helpful and things like that. I'll be literally very happy to read out all your comments out there. And yes, if at all you have any doubts, you can ask me now. Nisar Ahmed, ma'am, good afternoon, ma'am. I'm really so thankful. Your class helped me so much. You're the best teacher. Thank you so much. And Kamakshi also, thank you so much, ma'am. It's really helpful. Thank you, dear. Okay, ma'am, 42 question I have written hemophilia, alpha thalassemia. And also, I told you, dear, sickle cell anemia is not affecting chromosome number 16, because in sickle cell anemia, beta globin chain is affected, and beta globin is on chromosome number 11. Right? Yes. Not 16. Ma'am, I wrote about mutation, stability and replication for 2 marker. Mutation, stability and replication for 2 marker. Okay. Okay, 1 point has been missed, right? Hopefully, I really hope that you get marks for that completely. Will they cut the marks? They should not be cutting the marks. They might cut portion. If at all you explain the hemophilia and thalassemia well, and you explain why chromosome number 16 is also getting affected, you should be knowing about that. Ma'am, can you tell 44th B option? 44th, vector should not have more than 1 cloning site. Is that basically BAM-H1? Suppose this particular, the question means that if at all this particular restriction enzyme has multiple site, over here BAM-H1 will make cut over here. Suppose it has a site over here, it will make a cut over here also. If it makes a cut like that, what is going to happen? The plasmid itself is going to get fragmented, no? That is why it should not be like that. Okay? Ma'am, I lost 1 mark in MCQ question number 10. Oh, it's okay. Sisha, ma'am, I literally watched your master stroke video in 2x speed for concept clarity yesterday. I hope that worked. That worked well for you. I wrote the genetic material should be self-replicated, should be able to go for slow mutation, and it should mutate a slower rate as well. Goku. Okay. Okay. And you have mentioned about stability as well. You should be getting marks, dear. I literally hope so that you get the entire marks, yeah? Yes. Guys, do you have any doubts, or shall we end up this particular live? I think it's time to end the live, yeah? I only explained about hemophilia. Okay, dear. Probably we would have to go ahead and explain a bit more. Ibrahim, ma'am, I hope I get 95 plus marks. You will. MCQs were tough. No, dear, MCQs were actually easy. They were not tough. There are 3 confusing questions for you guys, and if you had attempted the live before or probably heard me before, I have told about the plasmid, the PCR question. We told this is the mistake that we usually do. You have to take care, and yes. But okay, it happens. Okay, what has been done is done. Now go and rest, enjoy your life, because your examinations are super over. But do not be worrying about anything, because yes, do not be worried about anything about your results and stuff. And please be back with your preparation for your KCET or NEET or whatever is there after some time. Okay, once you're ready for that, yeah. And ma'am, we will miss you a lot. Oh, that's so sweet of you. Even I'll miss you. Okay, I'll literally miss you. And make sure you're there for KCET examination if at all there is someone. I'm losing only in statement MCQ. Yeah, okay. Trisha. Perfect. Mama, Hiba says, I wrote hemophilia and alpha thalassemia properly. Then you will get marks there. Don't worry. Okay? Beta galactosidase, 44A. Beta galactosidase had no role to play over here, dear. So we cannot write about that. Okay? So cool, my dear children. This is what I had to tell you. And as I said, if your paper went well, and if you feel that it's okay to comment down, definitely comment down in the comment section how it went and things like that. You will do live for KCET, definitely will be doing that. Okay? Thank you for your efforts. Thank you so much, Ibrahim. Ma'am, because of you, I got more maths. Thank you, Jennifer. Thank you so much, Jennifer. Okay. Ma'am is beautiful with brains. Oh my God, thank you. Ma'am, I think I'll score 1770, but I get 69 and 70. You're still doing good. Sunita Stanley also says that thank you so much. You're most welcome. Muslim says thank you. Love you, ma'am. Love you too, Jennifer, Muslim, Trisha, everyone. Thank you so much. And guys, please take care of yourself. As I said, we'll see. Okay. And placenta is functional structural unit of the uterus, right? Yes, it is the functional and structural unit found in the uterus during pregnancy, and then you can tell that or how it is formed and stuff like that. Okay, that is chorionic villi, the uterine tissue and things like that. Humaira Begum, thank you so much. Welcome. I thought I would lose marks for writing extra sickle cell anemia. I hope you do not. Okay, because you have written everything perfect. I hope you have written in order hemophilia, then alpha thalassemia. So whatever is extra, they will just cut it off. Thank you, ma'am. Love from Belgavi. Thank you, Science and Stilly. Hiba also, most welcome. So guys, I'll end up the live, and ma'am, for 43rd A, I have written 3 and 9. So I told the answer, so just check with it, okay? I think that would be wrong somehow. Functional and structural connection between the, yes, it is a tissue, okay? We say functional and structural connection, that forms a functional and structural connection between this, yeah? Yes. So guys, I'll end up the live right now, and I hope your exams went well. And from tomorrow onwards, there will be sessions for KCET as well be there. And yes, that's all. See you, and tell me hi someday. Okay, here and there, if at all you see me somewhere, or probably you visit the YouTube channel, then let me know. Okay, your wishes are there. I'll be really happy to see that. And wishing you all the best. Please take care of yourself. Bye bye. Lots of love.